Excellent. Yeah, so even though these are a little bit out of order, we're just going to switch the first and second talk. But in any case, I'm going to be speaking on FracMinHash, which is a sketching method. So these 20 minutes, our agenda is to go over a little bit of the background, what the application area is, talk about the actual method, FracMinHash, analyze how it behaves with respect to error, relate it to average nucleotide identity, and then conclude. So let's start in the beginning and go ahead and talk about context. So my main application area is in metagenomics, that is a study of microbial communities from their sample DNA. So from a very high level, we have just a bunch of raw metagenomic reads from microbes that have been extracted from the environment. And we also have, associated with that, a large database of a bunch of assembled genomes of microbes. And we have on hand about 1.5 million of these full genomes. And the question that I have is, are we actually taking advantage of all the data in these sort of repositories at hand? So if you go and look at the different databases like GTDB, JGI Gold, or GenBank, it ranges from the order of maybe 500,000 to 1.5 million total reference genomes. But if then you go and look at the different tools that people use to be able to answer basic questions, like for instance, in my sample, which one of these genomes is represented inside of it? So tools like Motus, Metaflan, or Kraken, you'll notice that they use in their databases an order of magnitude or 2 or 3 less than the reference genomes we have on hand. There are many reasons for this, but one of those reasons is the fact that these the indexes that these different tools use can grow quite large when you're feeding it a lot of genomes. So what I would like to do is be able to leverage these reference databases as well as the amount of data that we have in their entirety. So to be able to compare these things in a very scalable fashion. So these databases are only going to get larger, our data sets are only going to get larger and more of them. So I want to be able to do it scalably and also be able to quantify the performance and accuracy trade-off. So to be able to do that, I'm going to be using something called a sketching method. So if you haven't heard of sketching approaches, a sketching approach is basically just some sort of data compression technique that still keeps whatever feature sort of preserved. So as an example, let's say that we have a really information-rich data point, like this picture right here, millions of pixels, tons of colors, et cetera. And we might recognize this as New York City skyline. Well, we could actually throw away a whole bunch of this data and still, like just with a simple sketch right here, notice, yes, that is still the New York City skyline. So that's a basic idea behind sketching techniques, and I'll be talking about a certain one. But where I'm going to use it is in the following. We have these big bunch of reads, and then from that, I'm going to extract one of these sketches that I like to think about as some smaller set of k-mers, or hash values associated with k-mers. And then when I do that with another sample, whatever I want to compare here, I can compare using these two as a proxy and be able to do it a lot more efficiently. So that's basically the idea. And for the rest of this talk, I'm going to be talking about k-mers. Most everybody here knows what k-mers are, substrings of length k. So like k equals 4, our substrings look like this. However, the method that I'm introducing is more generic. It's general purpose. It works on any set that you give it. So it also has applications outside of metagenomics and outside of genomics altogether. But for the rest of this talk, k-mers is what we're going to be focusing on. Okay, so what is this FracMinHash? Let me try to motivate it a little bit by intuition. So the way we're going to take a fingerprint of our metagenomic data, so we have all these reads. The very first thing that I'm going to do is get all the k-mers from all these reads. So after I get these k-mers, I'm going to get billions to trillions of these k-mers, especially depending on if I'm using a large k-mer size. And then I'm going to select some sort of compression level, some value s between 0 and 1, which is going to represent how much of the data I want to extract. So I'm representing it here. You may or may not be able to see the red points highlighted here representing the ones that I'm going to extract out of all my k-mers, and I'm going to represent that as my sketch. Then when I'm given another sample of reads or a genome, I'm going to play the same exact game. And very important here, the random sampling that I'm going to do is going to be repeatable in the following sense. So if a k-mer was selected in one of these, then if that k-mer also appears in the other data set, it will also get selected. And I'll show you how we do that practically in a minute. But I get this other list of k-mers as a sketch for the second sample. And then the overlap between these couple of sketches is going to be a proxy for similarity between the read sets. Okay, so now for the actual rigorous definition. First thing that you do is select a compression factor, select an S value between 0 and 1, how much compression you want. And then we have a hash function. This hash function is going to uniformly randomly map k-mers to an interval 0 to h. H there is the maximum value of this hash function here. And then our FracMinHash sketch, which, by the way, this sort of idea that I'm going to present, lots of people were sort of coming up with this similar sort of thing at the same time. So we all got together on Twitter to figure out what are we going to all call this, because we were calling it scaled minhash or fractional minhash or different things like that. And so we collectively decided FracMinHash, and you can go follow the Twitter thread if you want. But basically, it's going to be, this here is a sketch associated to a set of k-mers. And it's going to be the set of all hash values of those k-mers whose hash value is some fraction s below the maximum value of my hash function. So pictorially, what this looks like, I got a bunch of k-mers floating around. Here's my number line. In this case, my hash function goes from 0 to 100. So h, that maximum value, is 100. And then I can go ahead and fix my compression at 20%. I want to select 20% of my data. And then I just start mapping with this hash function. And I map my k-mers. And then I only keep the ones that fell below my threshold. And that there is going to be my sketch. Okay, that's all fine, cool, and good. As I move my S up and down, I can get a larger representation, more fine-grained representation of my sample, or a smaller one, more coarse-grained. But one of these sketching approaches is only helpful if you're actually approximating something that's useful. So in our case, we can prove in this manuscript that given two sets of k-mers, the ratio of the sketches actually approximates two kinds of set similarities, the containment index and the Jacquard index. So in particular, if you take the cardinality of the intersection between two of these sketches divided by the cardinality of one of the sketches, multiplied by this mysterious thing on the right that I'll mention in a bit, then in expectation over the hash function, you get an unbiased estimate of the containment index. And you can play that same game with the Jacquard index. You just take the intersection over the union of these hashes or these sketches, and then you get an unbiased estimate of Jacquard. So we can quickly estimate set similarity. One interesting thing, oh yeah, I'm going to focus on containment index here, and I'll explain in a bit why. Cautionary tale, turns out that FracMinHash is a generalization of a hashing technique called mod hash that was introduced in the late 90s. Basically, it's one that uses scale factor that's 1 over a natural number. From that paper, using modern notation, there was a claim that said the value, this expression, is an unbiased estimate of the containment index. And then you go look at the paper, and the proof says, the proof of this claim is straightforward. Unfortunately, it's not actually true. It's not straightforward, and it's not unbiased. So it turns out that, so after we did our little one-page proof here, there's this little bias term that sits here, basically accounting for the fact that you could get unlucky and none of your hash values fell in that interval from 0 to s times the maximum. So if you account for that, it's easy enough to take care of, but there you go. Great, so we define this thing. We know that it relates to set similarity. Now, how does it behave in terms of realistic biological noise, error, etc.? There are basically two sources of randomness that we have to consider. First one is the random sampling, because remember, I'm using a hash function, so that's introducing some bit of randomness. And the second one is inherent biological noise. And this could be mutations, it could be sequencing error. So let's take these one at a time. So for the very first one, with respect to the random sampling, we can prove that these FracMinHash estimates of the containment index are nicely behaved. In other words, they're asymptotically normal, which gives us this nice behavior. Yeah, as we change our hash function, it's just going to, in a controlled fashion, go around its mean. Similarly, there's concentration bounds that I can prove. I can show that the likelihood that your estimate exceeds some error is small. So it's high probability that you're accurate, basically. So, in other words, that lets me quantify the error. So as I change my scale factor here, you can see how the error is going to decrease. And this is quite helpful, especially for users, because I can give you accuracy guarantees. So you give me an S value, and I can tell you what set sizes you can approximate well and with what accuracy. I can also warn a user that if they input some parameters that they might get poor performance. And in real worlds too, in practice, we get some good accuracy. So you might be familiar with MASH, one of these other sketching approaches for set similarity. So here is some simulated data with the true containment index on the x-axis and the predicted on the y. And in red is MASH's containment, and the blue is the FracMinHash containment. This is a situation that highlights where FracMinHash does well when you're comparing two sets of very, very different size. So MASH, which uses MinHash, doesn't do so well when you're looking at sets of very different size, as you can tell by the huge variance on these, whereas FracMinHash does quite well. Okay, that was one source of randomness. What about the other source of randomness, the inherent biological noise? So this is the fun part. We have to have a model of error. What sort of noise are we dealing with here? So I'm going to use an independent, identically distributed Bernoulli model for my mutations, which is just a fancy way to say I'm going to flip a coin that comes up head p% of the times, nucleotide by nucleotide, and anytime it comes up head, I'm going to switch that nucleotide. So I have my sequence, I apply my mutation process, and you'll see here I have highlighted in red two of the mutated nucleotides. Now let's see how this sort of model of error interacts with my k-mer sets. So if we look at the k-mers for my unmutated sequence and look at the k-mers for my mutated sequence, notice that the mutation there propagates through multiple different k-mers. And if you look at it carefully, if two k-mers overlap, there's a correlation. If one is mutated, the other one has a higher likelihood of being mutated as well. Whereas other k-mers that don't overlap, a mutation in one has no effect on the mutation of another one. So theoretically, this turns out to be an M-dependent sequence of random variables, and so you can prove these big old scary theorems about central limit theorems, but I won't go over that because that was the subject of a recom talk a couple years ago. But in other words, all that to say, it lets us derive some statistics from it. If you look at the number of mutated k-mers, we can look at its expectation and its variance, as well as confidence in hypothesis intervals. Okay, how does that help us with FracMinHash? Well, let's go ahead and do both sources of this randomness at once. So let's go ahead and see how these mutations affect our FracMinHash and containment index. So here I have two identical sequences. And I'm going to have the k-mers, I'm going to color them differently, and you'll see why in a minute. But these two right now are the same exact set, so they overlap completely. But now, if I introduce a mutation into my second sequence, like right here, notice it affects multiple of my k-mers, and all of those get kicked out into one of the symmetric difference between here. And so you sit and stare at that long enough and you say, oh, hey, look, turns out the containment index is 1 minus just this fraction of mutated k-mers. Because the number of mutated k-mers is exactly how many are getting kicked out into this symmetric difference area. Awesome, so that's all good. So now I can take expectations over both my random sampling as well as over this mutation process, and that allows us to get the following. Expectation over both this source of randomness is this really nice, pretty formula 1 minus p to the k. What is that p? That was our mutation rate. And so with this, we can also do higher moments and be able to get things like containment or confidence intervals. You give me a mutation rate, I can tell you how big or small the containment index is going to go. But why are we going to all this effort to do all these expectations and things like this? Well, because it turns out this is related to ANI, average nucleotide identity. So as a quick reminder, ANI is the ratio of matching versus all characters in two strings. So for example, here are two strings, 50 nucleotides, all but 2 of them match, so ANI is 13 over 5. So ANI turns out, ignoring indels, is exactly equal to 1 minus p, that mutation rate. So now I have an exact way, using these FracMinHash sketches, to estimate ANI. And of course, people are interested in ANI because it allows us to do things like differentiate between species and microorganisms. So on both simulated data, where here the true ANI is on the x-axis, and or I guess 1 minus the ANI on the x and the prediction on the y, you can see that if you have very small mutation rate, both MASH and FracMinHash agree and are quite accurate. But once we start raising that mutation rate, you can see that MASH starts to overestimate it. So hence the title of this talk, you can do larger evolutionary distances with FracMinHash. On real data as well, we see the same thing. Here, FracMinHash isn't doing as well, probably because it assumes a model with no indels, which indels, transversions, various things actually happen in reality. So if you go and you look at real data with other ANI estimation tools like FastANI, PyANI, MASH, etc., some of these tools even say, hey, don't even bother using this tool if your ANI is below 0.8. And we can actually see that here because, yeah, these tools aren't actually returning anything. Turns out FracMinHash can estimate down to these low ANI rates. And if you're looking at very close ANI rates, it doesn't really matter what tool you use, they all basically agree. We can also, with this theory, let users know to be careful with the parameter settings. So right here, I have a heat map of both the k-mer size and the probability of mutation. The darker it gets, our containment value gets smaller. So moral of this story is if you're trying to reach at very far evolutionary distances, you better be using a small k value. Because as your k value gets bigger, then your containment index is going to go to 0, and there's no luck in trying to estimate that. A few other things that I wanted to touch on. You can get cardinality estimates. So if you want to know the total number of k-mers, you can get those straight from FracMinHash sketches. I mentioned that Jacquard, we weren't going to focus on that one because while it is true you can get mutation rate and ANI estimates from Jacquard, it's not asymptotically normal, so you can't get confidence intervals and things like this. One interesting thing, you remember when I mentioned these statistics of the mutation process and the number of affected k-mers? That took like 4 faculty members 6 or 8 months to be able to get like just the second moment for the variance. So shout out to my grad student Mahmudur who came up with a dynamic programming algorithm to compute arbitrarily high moments. So any moments of the number of affected k-mers under this mutation model can now be done. It runs in quadratic time, so it's probably of limited practical use, but very interesting theoretically. Extensions that aren't included in the paper include if you take these sketches and associate counts with them. So I have my sketch over here and the count of the number of times that those k-mers appeared for both two sets. And then I look at the cosine between A and B. That actually approximates the cosine similarity in the k-mer spectrum as well. So you can get cosine similarity from FracMinHash sketches. There's no reason you can't play this game with amino acids either. So you can get average amino acid identity. And hey, that means that you can get DNDS, number of synonymous to non-synonymous mutations, and talk about things like purifying selection, neutral selection, et cetera, from these FracMinHash sketches. And lastly, this is not my work, but I thought it was kind of cool. So Luis Erberg, guy whose PhD thesis I was on, indexed every single metagenome that's in the sequence read archive and made FracMinHash sketches from them. 500,000 data sets, was able to compress it down by 2 orders of magnitude. This allows you to answer some really cool stuff. What other metagenomes appear in other samples in the short read archive in a very short amount of time. So all of these formulas are implemented in SourMash. We worked with Titus Brown to put in these formulas into their code. Please go take a look. Comes with nice databases. A lot more I'd like to talk about, but I'll conclude with friends and funding, collaborators on this project, and thank you to the NSF. So thank you for listening.